In this video, I'm going to discuss the light-dependent reactions of photosynthesis. And of course, these reactions, as the name says, can only happen during the daytime or as long as the light is shining. We're going to focus on the four main events in this phase of photosynthesis, namely the primary photo event, the charge separation, the electron transport chain, and chemiosmosis. So let's start here by looking at what happens during the primary photo event. Light energy moves from the sun and it forms a form of potential energy, of course, but more so kinetic energy as it moves from the sun in packets called photons. And the light energy is absorbed by the chlorophyll pigment in a plant that is located in the thylakoid membranes. And we see that chlorophyll B, which is in fact the helper molecule, absorbs the sunlight, and the electrons in chlorophyll B become excited. As soon as that happens, those electrons start to move about and they jump from chlorophyll B to chlorophyll A, which is located in the reaction center of photosystem number 2. Now, chlorophyll A is the main chlorophyll pigment in a plant. So when the electrons in chlorophyll A become excited, they will start to jump around, of course, and eventually jump out of that molecule. And here we can see that path that the electron is taking, jumping out, and it will go next to a molecule that is called plastoquinone. And it will reduce that molecule. Now, plastoquinone is a movable carrier that can take this electron in the thylakoid membrane to the next electron carrier. But let's just pause there and summarize. So during this primary photo event, the light energy hits the chlorophyll molecules. It is chlorophyll B's electrons that jump to chlorophyll A, and from chlorophyll A, they exit out to the first electron acceptor in the chain of events. Now, before we discuss the electron transport chain, there's something that happens here. We'll see it is referred to as charge separation. And this is why we need to water a plant, of course. The water molecules will be split by an enzyme, a water splitting enzyme. And the water will lose electrons, of course, become oxidized, and it will form half a molecule of oxygen and two protons, hydrogen ions. And so these electrons replenish the electron hole that exists in chlorophyll B because, of course, it had electrons move from chlorophyll B to A. So there's an electron hole or deficit in chlorophyll B. So this is the charge separation step where the water molecule is oxidized. And now we can get back to our electron transport chain. We'll see after photosystem 2, the electrons, of course, reached plastoquinone. And next, those electrons will be carried to a hydrogen channel. And this hydrogen channel here can pump hydrogen ions through it into the thylakoid space. So it's coming from the outside, of course, which is the stroma of the chloroplast, and it is pumped into the thylakoid space. This hydrogen channel is the B6F complex. So let's write its name over here. And the pumping of the hydrogen ions through this channel will create a high gradient of protons here within the thylakoid space. And that is important because we're going to see later that that will be used for ATP production. So let's go back to where those electrons were. Of course, they are now traveling here through the hydrogen channel and reaches our next movable electron carrier here in the thylakoid membrane, and that one is plastocyanin. So from there, these electrons will next go to the second photosystem. They will reach photosystem 1. By this time, the electrons are slowing down, they are losing energy, and it's a good idea to re-energize them here, and that's why we see another photo event here as we see more light entering here and exciting these electrons. We'll again have chlorophyll B energized, and the electrons will jump out of chlorophyll B to chlorophyll A, reducing chlorophyll A in the reaction center, and then the electrons will jump out of chlorophyll A and to the next molecule following this photosystem. That molecule is known as ferredoxin. A complex here in the membrane that can accept the electrons, and so these electrons will be ferried or transported next to NADP reductase. So when these electrons reach NADP reductase, they will be transferred to NADPH. NADPH will pick up these electrons and become reduced. It is NADP plus that in fact picks up these electrons and forms NADPH. It also picked up a hydrogen ion. So that completes our electron transport chain, which is all the events that occur up here in the thylakoid membrane. Now step 4 follows this, and this is known as chemiosmosis. So we said that this high gradient of protons that are existing here in the thylakoid space is important. If we now pump 3 of these hydrogen ions through the ATP synthase pump, we can produce 1 molecule of ATP. So for every 3 that pumps through this ATP synthase, we can make 1 ATP molecule. So we're using a proton gradient, and we are seeing a type of oxidative phosphorylation occurring here, but it is because of this proton pumping that we refer to this process as chemiosmosis. So what we have here is 1 ADP picking up an inorganic phosphate, and it forms a molecule of ATP. The enzyme is ATP synthase. So now we see that we have our output of ATP, we have an output of NADPH, and of course we did make some oxygen. Our products, the oxygen will leave out of the plant. It exits through the leaves of the plant, little openings on the bottom of the ventral side of the leaves called stomata. And of course other organisms can use the oxygen for their cellular respiration process, but some of it will stay in the plant, and the plant cell will also undergo cellular respiration. The NADPH and the ATP next go to the dark cycle. And in the dark cycle, or the light-independent reactions of photosynthesis, we will see how the glucose is produced in that cycle known as the Calvin cycle.